Hi, this is Miss Litton, and we are getting ready for what exam? When is the AP exam? May. Okay, so May, what is it? 11th. What's today? 12th. So how many months away is it? 2 months. How many units do you need to be master of? 8. Yes, and all the science practices, right? All the labs we need to be masters over. So remember, what I do in this little 7th period, don't go, oh, Unit 1, check, okay, and scratch it off your list. Because you're going to need to go through that and look at it with some detail. Personally, what I would use are those master hyperdocs, okay, because I can go over a lot of things in class, but those lists at the beginning of the master hyperdocs, where I tell you this is 1.1, 1.2, all of those, yes, those are what the College Board says you have to know. So what I did is I looked at the list that the College Board gave me, and then I made my lessons match those. So that's why I lectured on what I lectured on, okay? Then they have what's called these enduring understandings, and those are at the bottom. And they can, remember, they're not going to see, remember the big ideas, the 4 big ideas? That's what they're going to be testing you on in each of these sections. And remember, pay attention to the top of those hyperdocs where I tell you what percentage of the test is over that material. Because whatever time you spend, it should be appropriate to what your test is going to be on. All right? It is not a mystery what's going to be on the test. There will be a ton of things you study that will not be on that test because they have a finite number of questions. Right? They're only asking you 6 essay questions. Think about that. I can ask you 7 or 8 on 1 unit. They're going to ask you 6 on our entire school year. So you may be #disappointed that you knew something really well and they never bothered to ask you that, and something you didn't know very well, they asked you a ton about. So what you have to do is prepare for everything by looking at every single one of those. Be scientific about your study. So you need to go through Unit 1 and read every single thing they say they're going to study on. And that's why I made it so it's like a checkbox. So you can go through and say, check, check, I learned this, I remember this. If you don't know what it is, get into our group shared notes, delve a little bit deeper. If you want a little more help, go to the Khan Academy links that I provided. You want a little more help, go to the Bozeman. Those are short little units where you can review that way. Want a little more help? Go rewatch the reviews that I posted just on Chapter 1 or just on Chapter 2, just on Chapter 3. Okay? You've got to develop a system and a plan. If you avoid it, it will not go away. All it's going to do is increase your anxiety because you haven't started yet. Okay? If for some reason school closes, okay? I hope it doesn't, but I want to do what's best and safest for all of us. Yes? You understand that? You guys are set. Because what are you going to do? You're going to go back. You have links to every lecture I've ever given, right? You have links to reviews. You can prep for that AP exam without me. I'm just here to get in your face mama bear style, okay? And try to get you to do it. What if I'm not here to review or teach you the final unit, ecology? Do you have access to that? Yes, it's on your hyperdoc. Will I still lecture through? Will we figure out a way to do that? And will I live stream it? Or you know that I will, right? So you will be fine. But what you can't afford to do is ignore it. If for some reason school closes for 2 days and we figure out how to do in-servicing, or they say we're not, you still want to take that AP exam. If they're giving that AP exam, you are taking that AP exam. You've worked long and hard for it. They have to figure out a way to make sure you can take that exam, right? So you study for it. They're not going to cancel that, right? They've got to figure out a way to make that happen. Yes?
So if it's not on the master hyperdoc, does that necessarily mean that we shouldn't study it? Or should we still review?
What you want to review, here's the tricky part, okay? Remember how now we have no systems, right? Systems don't have to be on the exam. But I've taught you systems, right? I've taught you systems because those are illustrative examples. So they may not have the immune system. You won't find it on there. But you find it because I use that to support, if you recall, cell signaling. How one cell can communicate through another, either through direct contact, when we looked at MHC1s and MHC2s, or through a chemical signal. That chemical signal could be interleukin or interferon, could be histamine, right? I dipped into the endocrine system a little bit so you could understand that's another form of chemical signaling. So I couldn't really teach you chemical signaling unless I taught you a few systems. So there may not be something specifically on the immune system listed from the College Board, but these are what's called illustrative examples. So what they might do is say, give me an example of chemical signaling. You're going to dip into your wheelhouse of all the different things we've talked about, and you're going to come up with your example. So you want to have an explanation for why that example works. Why that example works. Okay. Other questions or concerns about that? We're not going to be afraid. We're just going to make do because we're resilient people here in Oak Park, right? We have fires, we have all kinds of things. We know how to overcome. So if that happens, we will figure it out. Having anxiety in your life, is that going to help you in any way? No, not unless you're running from a bear or you're getting ready to compete in a sporting event. So do not scare or wig yourself out. We will figure it out. You are all young and healthy. You and I are all going to get the coronavirus at some point. We just don't want to get it at the same what? Time. Right? We don't want to get it at the same time. That's why we have to have precautionary measures. That's why they close the play. That's why, well, the rain is taking care of some of the sporting events. But that's why those things are happening, because we don't want to get around other people that don't use those precautionary measures. What's the number one precautionary measure I want you to use?
Wash your hands.
Wash your hands before you eat, before you touch your face. Okay? Wash your hands. That's the number one thing you can do. If somebody coughs around you, what do? Back off. Get away from them, right? Get away from them. Are these desks clean?
No.
Absolutely not. So don't touch your face if you've touched anything that anybody in the world can touch, right? Go to your room, clean your room. If you touch anything in your room, touch your face, it doesn't make any difference, right? But you do not have control over every door handle. You don't have control over all the atmosphere in every room. That's why you don't want to be in closed spaces with a lot of people, because that could facilitate the transfer, right? That's all. We'll be fine. Maybe we won't close at all. I don't know. But more and more are toppling, so I'm wondering, maybe that's going to happen, all right? So let's agree to a time when I would live stream. If I live stream something to you, what would be a good time? Want to say 10 o'clock? Okay. Yes. What?
We have to be in our other classes during that time. So
Maybe.
no, we have to.
You have to?
Yeah,
You're probably just gonna make
Yeah, I would. Yeah, I'm not gonna. Yeah, I'm not doing that all day long. It's not gonna happen.
You would need to like after.
Well, we would record it. Yeah, I could do it in the afternoons if you wanted to. Yeah, yeah. Now, I could do it like maybe at 2:30. Okay, 2:30, and then we would live stream all APs at 2:30. Are you good with that?
Yeah.
Okay. Now, let's also agree that in Google Stream, Google Classroom, I'll keep you posted, because we may not need to talk every day. I don't know how that's going to be, but if it does, okay? But 2:30 is APBio, yes? If it's a school day. Okay? Yes?
Wait, is it every day or every other day?
I don't know. You want to do it every other day?
Like on odd days when
Do it on the odd days? We'll follow the master calendar. On odd days at 2:30, I will live stream to you. Does that sound good?
Yeah.
Okay, odd days at 2:30. Somebody better send me an email about that, because I'm gonna forget. And you're on it? Okay, boom. Live stream 2:30 on odd days. Okay, we're set. All right, anything else out there you want to talk about? Yes?
For the labs, if school closes, are we just like not doing the labs, or?
Yeah, because I can teach you anything on those labs without ever doing that. Okay, so just be knowing that. Okay. Yeah. The only thing I'd be super bummed about is if you don't get to do genetic engineering, because that is awesome. It's just a very cool lab. But on the upside, we're like ahead of things. We're like ahead of the lot. So we're in a good place, because we only have one unit left. Some schools are just starting natural selection right now. I know, because they're telling me on TikTok. Okay, and they're like, our teacher taught us ecology in one day. Okay, you know. sad face. I don't know what to tell. All right, you ready? Anything else? I'll answer any question. I'll tell you no lies. What? Oh, yeah, damn. Well, it's okay. Yeah, there's 20 minutes. Okay, that's okay. Well, we're just going to start. So, your big ticket items, okay? Your big ticket items, a view to life, basic chemistry, and the chemistry of organic molecules. Let me just jump to chemistry of organic molecules, and then I'm going to work, okay. And then I'm going to work my way back. Chemistry of organic molecules, okay? Let's just jump. Boom. Remember this? These are your 4 important organic molecules. You know the name of the polymer is in your first column, right? The second column is telling you the monomer and little reminders on what it forms, what your building blocks are. And then on the right, I gave you all the polymers, okay? That is just straight up, you just need to sit down and review that, right? You're all at different stages at that. There are practice questions you can do on that. You just need to know those, okay? All carbs are built out of sugars. They are either used for structural or for energy. That's what all carbs do, structure or energy. Lipids have 2 big categories, fats and steroids. Okay, fats, you could think of things like phospholipids, right? Phospholipid bilayer, you would want to remember that. Fats could be used for energy. And then you talk about steroids. Steroids are a hormone, right? There's 2 kinds of hormones, protein hormones and steroid hormones. So you want to have examples of what they are and have that in your wheelhouse. Proteins. Proteins are built out of amino acids. You know that's in muscles and all that, but things like enzymes, those are proteins. Antibodies, those are proteins. So those would be examples. And remember, proteins are always very, very what? Specific. We also know about proteins, they can be what? Channels, what? Carriers, what else? Cell recognition, receptors, enzymatic junctions. Those are just in the cell membrane function, right? Just in the cell membrane, those are all the functions they can do. Okay, nucleic acids built out of nucleotides. You know about the different kinds of nucleotides, right? You're either going to be a DNA nucleotide or a RNA nucleotide. You know how they're same. You know they're different. You know about the central dogma. You know the central dogma says DNA, RNA, proteins, and then we've analyzed this and we think, wait, it might be what? RNA to proteins and to DNA. DNA could be our storage when we look at the origin of it, okay? If they ask you the central dogma, though, you're going to say what? DNA, what? RNA, proteins. Okay. Now, we know DNA is our genetic code. RNA is all about protein synthesis. And ATP is 3-phosphate. So instead of just adenine ribose phosphate, it's adenine ribose phosphate, phosphate, phosphate. And we know ATP is produced through cellular respiration. What the best thing you can do for yourself is do not think of things in isolation. When you study Unit 1 and you see DNA, don't think of it just like polymer monomer stuff. Think about, what are all the things I know about DNA? What do I know about alleles? What do I know about how they code for traits? What do I know about how DNA is reproduced through mitosis and meiosis? What do I know about the cell cycle? Because when they write your AP exam, they are not going to write it based on units. They're writing it based on all things DNA. Okay? So that's where you need to be thinking, because otherwise when they ask you a question, you'll be like, that's not in this unit, and you may not remember your answer. So you've got to broaden your horizons when you think about each of these things. Okay? So that is the third part, was just knowing about, right, the chemistry of organic molecules. You already know things about like carbon and why it makes 4 bonds, right? Because we know carbon has 6 protons and what? 6 electrons, right? And we remember how those electrons are formed because of the basic chemistry. We know the first energy level holds how many? 2, 8, 18, 32. Back it up. The atom story. Atoms are made of, be with me. Don't think you don't know this because some of you missed this on quiz. Atoms are made of what? Protons, neutrons, and electrons. The protons and the neutrons are in the nucleus. Atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8, 8 is great octet rule. Right? Okay? So we know the atomic mass of carbon is 12, right? So we know it has 6 protons and 6 neutrons, atomic mass, right? But we know that if it has 6 protons, what are the charges on that? Positive. So it's got to have the same amount of electrons, so that would be 6 electrons. And if we're going to put them away now, how many are we going to put in the first level? 2. How many are left over? 4. Okay. Is carbon happy? No. What are the 2 ways to make an element happy? Fill up its energy level, whatever level it's at, or get it to 8, to the octet rule, right? Okay? So, in whatever way you need to get it to 8. Might be giving away, might be getting an electron, exactly, okay? So, carbon's not happy, so how many bonds does it tend to make? 4. Because it has 4 electrons and it needs what? 4 more. Do you think it's going to steal 4 electrons? No, that's a hard pull, okay? So, more than likely it's going to what? Share. What kind of bond is that called when you share? Covalent bond. There are 2 types of covalent bonds. Polar and nonpolar. Polar is when you share fair. Nonpolar, right? Polar is when you share unfair. Sorry. You're like, what are you saying, Linton? Remember, no food. 45 minutes. Whatever it is. Polar is when you share unfair, right? Somebody has a stronger, more electronegative. Name me somebody who's super electronegative. Oxygen. He's always stealing electrons, okay? So he's always pulling them closer. Where's a classic example of when he pulls it closer? Water. Creating what kind of bonds ultimately? Hydrogen. Because they have a polar, right? Polar covalent bond that it leads itself between other water molecules to have a hydrogen bond. Yeah? See how we're thinking outside the box here, okay? So then when we go and we share fair, nonpolar covalent, that's what carbon tends to do, because how many bonds does it need to make? 4. So it's not pulling any more on one than the other, okay? Organic molecules, okay, are carbon-based molecules built out of carbon. The chemistry, when we look back at the chemistry, wasn't planning on working backwards, but it's okay. When we look at the chemistry, that's when we look at why atoms behave the way they do. Because if we understand why atoms behave the way they do, then we understand how molecules behave the way they behave, right? And if we understand the molecules, we understand the cells that they built. Case in point, okay? Long carbon chains. Carbons with hydrogens in them. Long carbon chains. Okay? Those are called hydrocarbons. Okay? Hydrocarbon chains hook up with glycerol to make what? A triglyceride. Good. And then what's a modification of that? A fat. Okay? And that fat, that modification, what if we remove one of those fatty acid chains? We can make a phospholipid. We can remove a fatty acid chain and replace it with a phosphate group, right? We know phosphate is phosphorus with a bunch of oxygen, yes? Oxygen is very electronegative. It makes sense to us that when we look at a phospholipid, that the phosphate part is going to be hydrophilic, whereas the long fatty acid chains are going to be hydrophobic. That, understanding the way oxygen and carbon and hydrogen work, help us to understand how a phospholipid works, which makes us understand how a cell membrane works. You see the connection there? So that's why you're doing the chemistry. The chemistry is not listed, the basic chemistry that I taught you is not listed in the AP College Board. You know why? Chemistry is a prerequisite to APBio. So that's why I had to teach you a little bit of chemistry in that chapter so you would understand organic chemistry, so you would understand the biology behind it. So that goes back, Josh, to your earlier comment. If it's not there, do we not need to know it? Part of it is they expect you to already have had a year of biology and a year of chemistry before you took this class. Do you need to be afraid? No. What pass rate do we have? 94, something in there. That's pretty good, right? And you're competing against seniors in other schools. So will you do fine? Yes, if you review and study, right? You got to remember all this stuff. The stuff that I'm bringing up right now, like when I said DNA as a polymer, did you ever think about DNA replication and the enzymes? DNA what? Unwinds and unzips, helicase, and you have SSV, but you have a leading and a lagging. Why are they leading and lagging? Leading runs from 3 to 5. Lagging runs from 5 to 3. And our enzymes can only work in one direction because enzymes are specific. And enzymes are built out of proteins, and proteins are built out of amino acids, right? And so then we said, well, to get the party started, we need some primer. Who makes it? RNA. Remember RNA primase? Primer. One time on the leading strand to get the party started. Multiple times on the lagging strand, right? Okay. Then what comes next? Do you remember? DNA polymerase 3. Remember? Boom, 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 boom. Right? It works and it makes those short 1,000 to 2,000 base pairs called Okazaki fragments. Now what do we need to do? We still got primer in the picture. What are we going to do? DNA polymerase the 1, because he is the one to save us. Primer, one time on the leading, right? Primer, primer, primer. I'm only jumping around because I'm thinking, what if this last time I see him? Okay, primer, primer, primer. Then I remove that lagging, right? Multiple times I have to remove the primer. Then what do I need to do? Ligase. Why? To seal the phosphate sugar backbone. Because you're going to form hydrogen bonds between the nucleotides, so you don't need to worry about that, right? It's the backbone that you have to worry about. Then these are going to what? Twist up and form sister chromatids held together by a centromere, separate during anaphase. Anaphase of what? Mitosis. What's the first step of mitosis? Prophase, the chromosomes become visible. APBio in 5 minutes. The nuclear envelope and the nucleolus disappear, radiating microtubules, pastors. They meet in the middle at metaphase, away at anaphase, two nuclei at telophase, cytokinesis. That is mitosis when we are making clone cells for growth and repair. Yes? But if we're getting ready for sex, if you are a sexual organism, you're going to do what? Meiosis. In this case, you have to take a 2-end cell and make it a 1-end cell. Good job. Okay, so let's talk about this. Still, what stage do you replicate the DNA in? What is it? S stage of interphase. So that has already occurred, right? So we're going to do meiosis 1. What's the first stage? Prophase 1. Okay, so show me your chromosomes because the chromosomes become visible. Now, how many chromosomes am I showing you right now? 2. 1, 2. Each chromosome is made out of sister chromatids held together by a centromere. This is referred to as a homologous pair because I might have gotten this one from mom and this one from dad. If both of these for eyes say big B, am I a homozygote or a heterozygote? Homozygote dominant. And my eye color will play out. This is my genotype. My phenotype would be my eyes are brown, right? I would need little b, what would that be? Homozygous recessive. And if I was talking Hardy-Weinberg, what would little b be in the equation? Q squared. Anybody with brown eyes would be what? P squared. Plus 2pq, right? Those would all have brown eyes. But some of them would be homozygous and some of them would be heterozygous. If I just look at my gene pool and look how many brown alleles out there, what would that be? P. How many blue alleles, what would that be? Q. Because P plus Q equals 1. And when I'm doing that, I'm studying population what? Genetics. And population genetics says if the allele frequencies change, then what is occurring? Evolution. What kind of things can cause those allele frequencies to change? Small populations, non-random mating, mutations, gene flow, and natural selection. right? So when we talk about mitosis and meiosis, the whole thing about fitness and getting your genes passed on, meiosis is making those haploid cells of the DNA that has been selected for through natural selection, unless it was chance, and if it was chance, what is it? Genetic drift. What are the two kinds of genetic drift? Automatic and founder's effect. Good. So back to meiosis. How many chromosomes do I have? 2. How many chromatids in each one? 2. Okay, this one has 2 chromatids and this one. Are these the homologous pairs? No, these are the homologous pairs. If this came from mom, this came from dad. So meiosis 1, the chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, after synapsis. What can happen during synapsis? Crossing over. What can this increase? Variation. That's what, who couldn't explain? Darwin couldn't explain that because he didn't understand the chromosomes, right? So that can increase variation as long as they're non-sister chromatids exchanging. Then what do? They meet in the middle at metaphase 1. Now when they meet, they can meet in the middle at metaphase 1. What is that called? Independent assortment. Because each homologous pair assorts independently of all the other ones. Yes? Then we go away at anaphase 1. Who's going away? Homologous pairs. And then we make 2 nuclei at telophase 1, cytokinesis. Okay? Now, technically, our cells are what? Haploid. Why? Because we separated the homologous pairs. Now we're going to do meiosis 2, starting with prophase 2. Can we do crossing over? No, we cannot. Why? Yeah, these are sister chromatids. Can we meet in the middle at metaphase? Just single file. They meet in the middle at metaphase 2, right? Because you're in 2 different cells. Metaphase 2 in 2 different cells. Away at anaphase 2, 2 nuclei at telophase 2, cytokinesis. Except if I'm doing oogenesis and I'm making female eggs, then I will do unequal what? Cytokinesis, because I'm putting all the nutrients in one large egg. What are the other things called? Polar bonds. My gosh, I think we just did like 10 units right there. Now, so we have the chemistry. We also talked about the properties of water, and you can hover all the properties of water around those hydrogen bonds, right? We can look at the way it behaves due to that hydrogen bonds. Why would we want to know about water? Because our bodies are mostly made out of water. What do you call the diffusion of water through a semi-permeable membrane? Osmosis. Water must flow from the hypo into the hypertonic medium. Now, that normally works fine on a cell membrane. The only thing that might affect that is if you have a cell wall, because the cell wall will push back. And what is that called? Turgor pressure. So then we have those water potential equations. Do you remember those water potential equations? Of course you do. Water potential is a factor of solute potential plus pressure potential. Do you remember that? And we could calculate that. And in fact, if we wanted to make the pressure potential 0, then the water potential was only a factor of solute potential. Do you remember the equation for solute potential? Negative. I'm hearing it. ICRT. Do you have to memorize that? No. It'll be on your table, but you will find yourselves all the time when you're taking an AP exam, you're like, don't need it, don't need it. Sorry, hope you didn't miss too much. We were outstanding. Okay, so, I don't know, at 29 minutes.
I was like still.
We started at 2:44.
Oh, okay, Swedish. All right, so hopefully you didn't miss too much. We were reviewing water potential. Okay, oh good, I can work now. Okay, so the energy starts with the sun, right? I know, it's crazy. Okay, and then autotrophs can capture the energy from the sun and do what process? Photosynthesis in order to make sugars. And then animals can eat the plants or eat other animals, right? and then that's how they get their nutrients. And then the plants and animals die. Who works on them? Decomposers. Circle of life symbol. Okay. Now, here we go. So we plot out whether it's gaining or losing weight. Then we use a ruler and we make our best made line. This, I'm assuming it's lost all the water it can and it's just kind of bottoming out. So I would probably, make some line through here. Okay. And then I look to see where does it cross zero molarity. So what I would envision is they would give you data and you would plot it and then you on a graph and then you would have to make a line, okay? And then what you would do is you'd go, okay, so this is 3, so for this situation, let's say this is 0.25 molar. Am I making a good choice there?
Yeah.
Okay, 0.25 molar. So when I plot it in here, right, this right here is going to be 0.25 moles per liter, right? And then your R is 0.0831 liters per bar per moles per Kelvin, but you could just look that up, okay? It'll be on there. And then what's this right here?
273.
What is it?
273.
Yeah, it's 273 plus the temperature. That's Kelvin, okay? So when we did this experiment here, we would want to know what temperature did we do it at. So if they said it was at 22 degrees Celsius, you would have to go 273 plus 22, right? And then that would be this number. And then everything will cancel out except bars. And that's how you would calculate the water potential. Okay? So we just went through a whole lot of things right there, right? So hopefully those highlighted some things you remember. And then if there was something in our discussion you're like, I don't want to know that, what should you do? Go back and review it. Okay? Hopefully we have a lot of more days together. Just throwing that out there just in case. Yes?
Can you probably do quizzes and tests if you're out of school?
How could I possibly quiz or test you? I couldn't. I can only give you like formative assessment, so you can check for understanding. Yeah. Okay? I love you. Make good choices.
Wait, so